artificial insemination in small ruminants. In today's presentation, we are going to see about what is AI. Afterwards, we are going to see the factors affecting the conception rates, the advantages, disadvantages related to artificial insemination, the anatomical structure of the female sheep and goat, as well as their reproductive cycle. And when we are going to discuss about the synchronization methods, equipments, and techniques related to artificial insemination. When we talk about the background of artificial insemination, we must know why it is required. Why we are going to talk about the artificial insemination, why it has been so popular. As we all know, this method is so much popular in large animals, but nowadays it is burning in small ruminants too. Why? Because we know the population is growing day by day. According to the UN Population Division, the growth rate of India population is increasing 1% between 2010 to 30 and about 0.5% between 2030 to 2050. We can see the data showing the slow growth rate. Still, it is to be increased and projected to be 1.6 billion by 2050. So when we talk about the increasing population, the human need food. We all know each and every scientific study and science is done only by the benefiting the and sustainability of human race as well as the animal. So when the consumption of food increases by the huge population, it can be achieved by two ways. If we increase the number of animals, or we can increase the animal productivity. And we know that sheep and goat meat consumption is very popular. And according to data, we have been found that between 2009 to 2050, it is going to be rise by 117%. It is recommended by the Standard Authority of Food and Food Regulation that urban consumption would be increased by 249%, as well the per person consumption would be rise by 47%. So to nullify these demands, we have to increase the animal productivity, whether in terms to increase the animal or to increase the animal productivity. For this purpose, when we talk about the natural service, it is not going to fulfill this demand of human race. So we need the process of artificial insemination. Artificial insemination have various advantages. In upcoming slides, we are going to discuss it briefly. So one must know what is artificial insemination. It is the deliberate introduction of semen, which is a ejaculate, comes from male into the female reproductive tract, female body, for the purpose of achieving pregnancy, a successful pregnancy, though fertilization by means of other mating than natural services. So this is the concept of AI. Other than natural service, the human-made effort by introducing the semen into the female reproductive tract is considered as artificial insemination. And the main goal of artificial insemination is the genetic improvement as well as the control of venereal disease. And if the population of genetic will improve and the population of animal will improve, it will correspondingly satisfy the human needs also. When we talk about the artificial insemination, it is one of the most popular, widely used artificial reproductive technique. It is very much successful in large animals, but it is very much helpful in small and ruminants too. It is an assisted reproduction procedure where sperm is collected from male and then manually deposited into the female to achieve the pregnancy by passing the sexual intercourse. Here we need a semen, frozen semen in straws. It is made available by different institutes, and hand over to AI workers or the learned veterinarians to go into the field and do the artificial insemination. One must know what is the procedure of artificial insemination. When we talk about the procedure, first of all, the foremost, we need a good genetic trait male animal, buck, a male animal of apparently healthy and showing good merits of body scores. Then we are going to collect semen from it. In later slides, we are going to discuss about the method of collection of semen also. When we collect the semen from a desired male animal, then we are going to evaluate that semen, whether it is up to the mark to get into the female body or not. If the semen evaluation is checked, then we will further process to its preservation. For the preservation purpose, we need a suitable extenders as well as temperature should be regulated, because we know the semens are the living cells which are very much sensitive and fragile towards the stress in terms of temperature, storage, handling, any type of the secondary stressors. It is very much sensitive cell. So we have to take a lot of precaution for preserving it. Afterwards, we have to, as soon as the semen is preserved, we have to finalize the female. When we are finalize the female animal by identifying and prepare her for mating. Some purposes we use, some cases for collection of semen, we use dummy animal also. But in artificial insemination, we use a female who exhibit estrus cycle, who is in its estrus phase, showing the heat signs. We have to identify her and prepare. After preparing the female, we have to collect that preserved semen and thaw it properly. Thawing of semen means bringing to that semen to that particular required temperature in which it can be introduced into the female body. Afterwards, we will do the insemination. There are various techniques also. We are going to discuss about the various techniques of AI. There are generally 6 different techniques. Manual and laparoscopic. In this, we have 6 different techniques and many more. Then this is by insemination of semen in the female. We have to look over and monitor the female up to the successful parturition. This is all about discussing the artificial insemination process. Let's talk about the collecting semen methods. How we are going to collect semen. We are going to collect semen by artificial vagina, electroejaculation method, massage, by breeding bag, by the help of real vagina, and by digital manipulation. But among these all, artificial vagina is very much popular and recommended method, because in this the chance of disturbances in sperm is least. These methods of artificially collecting the semen, in spite of natural mating, have several benefits, several advantages. Like if the animal is in injured condition, having overweight, or any type of health condition, the male is any fracture or any injury, any type of things can be happen to male, and he cannot stand up and ejaculate. So we come to take the help of these methods, which are collecting semen methods. When we talk about the artificial vagina, it is a very common way to collect the semen from male animals. We can take But before artificial vagina, I would like to discuss about the 5 stages of collection naturally. What happens in the reproductive cycle? There, when male and female gets intended, they do the foreplay. And after foreplay, the erection of penis will occur in the male. And this erected penis will get inside into the female reproductive tract by the term called as mounting. And the mounting, the animal will ejaculate its semen into the female, and the restlessness or dismount will occur after ejaculation. This is the normal process. But when we talk about the artificial vagina method, it is a very common way. It involves very special design devices. Here we can see this is the artificial vagina having an insulated cylindrical body for collection of semen. The recommendation for artificial vagina method collection of semen, that area should be peaceful, it should be free from dust, and have protection against summer and winter's wind also. Then we have to put the teaser eave in a neck clamp and introduce the ram for short intervals until the ram mounts. The AV, this equipment is an intimation of the vagina of the doe. It is in market. The AV is available for different species in different shape and size according to the male requirement. It has 3 main parts when we discuss this. It has an outer cylinder made up of canvas and reinforced with rubber. Inner rubber liner, it is made up of soft latex, and the rubber cone, which is fixed at one end. Then we have to put our sterilized graduated centrifuge tube around 10 ml to one end attached to this open end of collection semen. One must important thing that the temperature, you have to maintain the temperature of liner. It should be around 42 degrees centigrade to 45 degrees centigrade. Then this AV is filled with hot water around 50 degrees centigrade to 55 degrees centigrade. This generally provides correct internal temperature and prevent the semen from cold shock. And the tube, collection tube should be warmed around 30 degree to 37 degree before collection. The tube should be lubricated with sterile jelly before filling the warm water. And then it is inserted into the penis. The pressure and temperature adjusted as we have discussed. This will sensitize to the male and natural mating experience will feel the male. Once the male ejaculates, the semen is collected, the collection machine, and attached to artificial vagina. In this way, we get the semen in graduated tube. There are certain advantages. What are the advantages in artificial vagina method? That it stimulates natural ejaculation. It is less stressful and more over natural to the method of collection semen. That's why it is very much preferred method. And about when we talk about the semen we get is of very good quality. It is having good quality semen with better motility as well as the morphological characteristic is also excellent in this method. It is widely used, well-established method for semen collection in various species. In small ruminants also it is most popular as well in large ruminants also. When we talk about the electroejaculation, we need stimulator. Electrical stimulator is required in this. It is a method in which semen collection is done by obtaining low voltage electrical pulses and applied through transrectal probe to stimulate the ejaculation. We have to prepare animal, whether you have to sedate or without sedation. Sedation is generally scientifically advised to minimize the stress of animal. But people also generally practice without sedation also. Then the special probe or stimulator is inserted into the rectum area of the male animal, and electrical stimulation is, here we can see in this figure, is applied. Of very low voltage electrical pulses should be around 10 to 15 volt output, it should be introduced, and afterward the semen has been collected and stored properly. One, you have to sure that the depth in for rectal probe should the rectal probe should be lubricated, and the depth should be around 15 to 20 centimeter to avoid the injury of rectum or rectal orifice in the males. By this method, the glans penis shows congestion and the correct stimulation of semen is done, and it should be collected in a pre-warmed sterile tube. When we talk about the handling and examination of semen, the semens are very sensitive. For cold shock, glassware for collection, handling must be clean, sterile, dry, warm, minimum 30 degrees of centigrade. And the work should be done in clean and enclosed area, protected against sun, dust, wind, insects, or some flies. So we get this semen and put into the water bath also for maintaining the temperature. It should be considered in this method that there should be less stress and pain. The quality is little much compromised, and sedation is one most the challenge comes, and the equipments and the skillful persons should be needed for electroejaculation. Once we get the ejaculate, we should monitor the characteristic for ejaculation. First, we have to see the concentration. In lab, we have to bring the semen and check its morphology as well as the characteristic. The analysis of semen we have to do. In this, first and foremost, we have to check the concentration of semen, whether it is up to the mark, excellent, good, or very good, or it is poor, the concentration. And we have to check it should be how much millions per ml concentration. Then second, we have to talk about the progressive motility. How the semens are moving progressively back forward, or we have to check its motility, whether the semens are moving forward or backward direction. It should be mass motility. There should be percentage mass motility moving forward. So we can suggest the semen is showing excellent quality grades. Then we have to see the acrosomal morphology. How much percent intact acrosomes are present in the semen sample? Then the volume, whether the animal is ejaculating proper volume of around 1 to 2 ml. In small ruminants, we get the semen ejaculate. Then we have to check the tail morphology, whether the tails are coiled or straight or having droplets kind of things, or they are swollen or shrink. So we have to check tail morphology. Afterward, we have to see the visible test, like we have to see the color, the thickness. By visualization, we have to check whether the color of ejaculate semen is okay. What is the pH? We have to check these characteristics before preparing semen for ejaculation. Now we are going to talk about what are the factors which affect the conception rates in animals. Though it varies from place to place. Some places the animals are showing very much good conception rate, where in other hand, some places showing poor conception rate. Why this? There is differences occur. So there are some points. Generally, I have shown in bullets. We're going to discuss about one by one what are the factors which affect the conception. Like first and foremost is the managemental aspect. If you are not going to manage your animal very well, ultimately the conception of female will hamper. The fertility rate will be decline. So in management, one must try to introduce proper ration to the female. Flushing should be done, like proper feeding, proper giving of less posture before parturition to the certain period of gestation is mandatory. The mineral mixture, the salt percentage, the supplements, the probiotics, these all factors plays very crucial role in nutritional aspect of animal for having good conception rate. If this will not checked properly, sure and shortly the conception rate will decline. Then we have to check whether the animal is free from parasites, internal or external. Is the animal is free from any disease, any chronic, any acute disease, any kind of infection? It should be checked. Proper medication, vaccination should be suggested to animal before and after ejaculation or insemination. The body condition, when we talk about, it's depend upon the nutritional status as well as the genetic score. Whether the body conditions and the body score is favorable for giving a normal offspring, this must be checked. What is the season? When we talk about the goats, these are short breeders. They require less daylight. The season should be proper as they are seasonal breeders. Season plays a very crucial role in the conception. If the season is not supporting the animal, there would be surely fertility decline. When we talk about the estrus detection, estrus can also varies. It should be checked very properly by the farmers and the owners. The marginal raiser should check the estrus detection. If it is not detected, one must synchronize it. In later slides, we are going to see about the synchronization process also. And lastly, what we have talked earlier, that one must check the semen quality because the seminal attributes also impact the fertility rate as well as the conception rate of in females. What is the once artificial insemination is done, then what are the advantages we are going to get? We are going to see, no doubt, the superior genetic elite offspring having the fine quality of genetic expressions. Like when we talk about the milk purpose breeds of sheep and goat, we will have a good amount of milk production. And when we talk about the meat purpose, we will have a good amount of meat also. When we talk about the having genetic purposes, genetic qualities, having meat and milk, good merits, we have ultimately we have future cost and benefits. We have for AI, we have future cost and benefits. AI is a method in which it is very cost friendly also. For the safety purpose, the disease spread can be checked and the infection rate can be overlooked. By AI, we can control kiddling as well as lambing window also. If it is not required, it depends upon the circumstances. We can check over. We can check the peculiar back smell which comes from the meat. We can also check that. We can also check. Uh, we have the housing as well as the pasture advantages by artificial insemination. We don't need much required spaces. The space for maintaining animal is also can be checked. And lastly, from the disease status. Now we are going to discuss about what are the disadvantages of artificial insemination. As we know, every coin has two sides. One is head and one is tail. If there is some merits, sure and sure there will be some demerits also. So we must know what are the demerits by artificial insemination. We will going to see like startup cost. The startup cost is likely to be rise as compared to the natural services. The managemental aspects. One must have to manage all the animal as well as the equipments very well. So moreover, this is a disadvantage for it. If it is not managed well, you are not going to have a proper AI result. Then the time. Time in terms of heat detection. If the female surpasses its estrus, then there is no use of artificial insemination because the animal is not receptive. So, proper heat detection, proper record keeping, it may vary from animal to animal that females can show various estrus length. And third, for artificial insemination, a skilled person or a worker is required. Without skill and proper knowledge, artificial insemination can't be done properly and the results will not up to the mark. When we talk about the poor sires, it is also when we get a straw from a low quality trait ejaculator, the artificial insemination will go down. Let's talk about before the artificial insemination procedure, we must have the understanding of female reproduction. In female reproduction, the female possesses several functions. We are very well to know that in female reproduction, we have certain merits that in female reproductions, we have ova provided in terms of eggs, which are the core for fertilization. The female reproductive tracts provide proper environment for fertilization. It nurtures the embryo as well as the fetus throughout the gestational length. Throughout the gestation length, the embryo is being safe and preserved inside the mother's womb. Afterwards, it delivers the fetus to exterior and the female will continue to feed the young goat. This is the lactational period. And in female reproduction, we are also seeing the proper behavior patterns like attracting male and mating with males. The female will approach males in its heat period and demonstrating the maternal functions towards the offspring, towards the lamb or kid. So these are the specific properties of reproduction of female. When we talk about the reproductive organ involved in reproductive system, we would find there is a presence of ovaries, which is 2 in number, situated symmetrical both in abdominal or pelvic cavity, in right and left side. There are 2 ovaries. Then these ovaries have tube-like structures which attach to uterus, that is called as oviduct. Uterus is a house for keeping worm, and at the end of uterus we are going to find the cervix, which is the passage for semen traveling, and after closure of cervix we would find vagina, and it will end up with the exterior wall called as vulva or os. When we talk about ovaries, it has 2 principal functions. First one is the production of ova, that is eggs. And second one is the secretion of female hormones. One is estrogen as well as progesterone. Estrogen helps in follicular dynamics and progesterone helps in maintaining pregnancy. And these two hormones are controlled by the anterior hypothalamic hormone, anterior pituitary glands, which secretes LH and FSH, and it is controlled by the hypothalamus. So this is the gonadal axis. When we talk about the fallopian tube, it is divided into three junction. Here we can see first one is into infundibulum, ampulla, and isthmus. This fallopian tube provides the site of fertilization and early embryo development before the embryo passes to the uterus took place here. The transport, helps in transporting ova from the ovary to the site of fertilization, which occur midway down the oviduct. Ampulla is the site of fertilization, one must know. And at the same time, the sperm cells move in other direction towards the ova from the uterus. Means two cell movements are going through this tube at the same time. The eggs are traveling towards uterus and the sperm which is coming from the semen ejaculation, this is coming towards the egg. Afterwards, when we talk about the uterus, it is a small muscular organ that provides protection as well as the nourishment for the developing embryo. It is consists of two horns as well as body. The uterus is having different shapes in different species, like it is biparotid, Y-shape, T-shape uterus. According to species, it varies. Having two different horns. In polytocous animal as well as the monotocous animal, when we talk about the polytocous animal, the pregnancy sustains in horns only. The inner lining of uterus is made up of button-like projection called as caruncles. And these caruncles are attached with the cotyledons, which is served by the fetus. And we found the placentome also. We found the placenta, which helps in transferring nutrients between the dough and the developing embryo take place. So the placenta also is stuck into the uterus for providing the nourishment to the embryo. The last, not the last, this is the cervix portion. This is the muscular canal portion. It is a structure that provides closer to the uterus of a doe, and it protects the uterus during pregnancy. Throughout the pregnancy, the cervix is playing very much crucial role in protection purpose. None other infection, no any external agent should enter the uterus and disturb the pregnancy. It is checked by the cervix only. During breeding time, this cervix also assists the movement of sperm from the vagina to the uterus. So it is act like a passage also and protects the uterus also. When we talk about the vagina, it is the site of semen deposition by the birth during natural mating. Natural mating, we have earlier discussed by the vagina method, we can also collect the semen. So some people in earlier days when their AV is not introduced or electroejaculation was not introduced, people used to collect semen through the vagina only. But it's not so much advanced and recommended method because the disturbance and deformity in semen can occur. Once deposited, the sperm, the cells are transported into the cervix and seminal fluid is either absorbed or it will be expelled by vagina. So this is the role of vagina. Lastly, when we talk about the vulva, it is the external opening of the female reproductive tract and it serves as an entrance for the penis during breeding, and at the time of parturition, it is the end of birth canal. Here we can see the goat external os. This shape, this T shape is showing the goat external os. When we talk about the cervix, of goat, we can see it is a long muscular structure. It, when we see it's some star shape, some duckbill, some crescent, some spiral or cluster or the bump shape we can found in this cervix. So the cervix is little more advanced in goats, but in sheep it is more advanced than sheep. This is the external os of sheep. And when we see the cervix, we can see here how the protrusions have shown. When we see anatomically dissecting the cervix, we see like this. The ridges are seen. Let's talk about other important point, that is the estrus cycle. What is a estrus cycle and how it differs from species to species? So it is a cyclic sexual rhythmic cycle. It is an interval which occur by a certain, which repeats by a certain interval of time. It is divided into 4 different phases: proestrus, estrus, metestrus, and anestrus. So before estrus, there is a period called anestrus or diestrus. Then we have estrus period. Basically, it lies end of follicular phase, when the all the follicles get mature and starts to rupture. Then the estrus period will occur. At this time, the ejaculation of ova from the ovary took place. The Graafian follicle will ejaculate the ova when the estrogen level highs, and we would see the estrus cycle. This time the ova is released into the tract, that is fallopian tube, and very much animal is appropriate or interested in mating. So this time the animal will show certain signs of estrus. And this is the estrus period. When it gets over, this cycle is, and then luteal phase will occur when the progesterone is dominating. Afterwards, the peak of progesterone get declines if there is successful mating. Then this peak will continue for progesterone. And if there is no successful mating occur, then this hormone gets down and again the estrogen will took place and the follicular phase will start, and another estrus cycle we will going to see. So the duration between one estrus period to other estrus periods is called as estrus cycle. And this cycle differs in different animals. When we talk in respect of goats, when we talk about goat or female goat, they are the seasonal breeders. They breeding season initiate by decreasing the daylight. However, individual does not have either longer or shorter intervals. Around 24 to 36 hours, they show that duration of heat. At this time, the animal should be inseminated. When we talk about the stages, as we have seen, there are 4 stages: estrus, metestrus, diestrus, and proestrus. The day cycle for estrus is 1 to 2 days. We can say 24 to 36 hours. Here we can see the characteristics, the final growth and ovulation. 1 to 2 pre-ovulatory cycles will take place. The sexual receptivity of those will occur. The mucus will become, cervical mucus will become thinner and more copious. Then the period of metestrus will occur. It around 3 to 4 days of cycle. In this, the functional organization of corpus luteum will take place. Afterwards, the diestrus of 5 to 18 days will occur, the proper luteal phase. Then proestrus, proestrus, proestrus for 19 to 21 days. The growth of pre-ovulatory follicles will occur in this phase. When we talk about sheep are different as compared to Goats, here we can see the estrus cycle of 17 days, having various phases of estrus. We can see the insemination timing. When to breed for best success? It is 6 to 28 hours after the onset of heat. From the from the 9th hour of standing heat. Standing heat is the point in which the doe heat cycle, when she is receptive to the buck, and the heat is the female reproductive cycle, from 9 to 21 hours. This is the time for excellent breeding. The animal is ready to breed. But 6 hours before and last 24 to 27 hours is also good time to inseminate. What are the signs we find in females for estrus? That is, it is a polyestrus and short day breeder. The animal reluctant to mount. Vocal voices are produced. The flagging of tail will be shown. The females are search out of males walking in the fence lines. Restlessness we will found. There will be loss of appetite. The production of milk will be declined. The female will start urination frequently. Maturation occurs. The vulva become swollen red and there will be mucus discharges. In the pictorial form, the same I have tried to represent all these signs of heat. When we talk about the sperm cells, these cells have in short, small ruminants, we have around average ejaculate of 1 ml, in which 3 millions we get per ml of sperm. When we talk about the straw, when we have half ml of straw, we are having around 250 millions of sperm, in which half have been lost during freezing, around 125 we have, and half after thawing. So at the time of insemination, we have around 62 million of sperms. Minimum need of 60 millions for pregnancy is needed. One must know this. How we are going to synchronize this estrus? This estrus can be synchronized by progesterone, implant or pessaries. Different dose rate and different implants in is recommended. By injections of PG of prostaglandins, PGF2 alpha is suggestible. We can synchronize by heat check method, the NC sync and the CIDRs methods also. This is the control device and combination of prostaglandin and FSH shots. When we check the synchronized method, simply heat check at the day 0 initial, we have to give injections of PGF2 alpha, and at then day 10 we have to repeat this dose, and after the animal is ready for at day 10 to 17 the breed is ready for insemination. When we talk about the NC sync, the same happens at the 0 day, but at the 7th day we have to give the injections for GnRH, gonadotropin releasing hormone, and at the 14th day we have to repeat the day 0 dose, and at the 17th we have the animal is ready to breed, or if required we can again give the GnRH. When we talk about the CIDRS, the day 0 we have to implant this, and day 11 and 17 we have to pull it out and administer FSH and breed the animal at 48 hours. For artificial insemination, what the equipments we required? First one is the semen, and this semen is kept in this semen tank, which is temperature, which have a jacket which is temperature sensitive, and the temperature is controlled in this semen tank filled with liquid nitrogen at minus 190 degrees centigrade. Then we have AI gun, the dipstick measures. This is the tweezers. This is the sheets. This is the plastic sleeve one must wear before AI. The thermostat for thermometer for thawing purpose, and the napkin. The straw cutter for cutting these straws, sheets, and this is the semen. This is the basic equipments which are required for artificial insemination in the field conditions. When we talk about the insemination, there are different insemination procedures, like there is intravaginal procedure, intracervical, intrauterine, tuboperitoneal, intratubal, as well as intraperitoneal, in which intravaginal and intracervical we can done manually by hand, and afterwards the intrauterine, intraperitoneal, intratuboperitoneal, and intratubular we must recommend the laparoscopic method by the invasive method by the surgical operation. In intravaginal insemination, we can see here this lady is doing intravaginal insemination by the help of this AI gun. This is a normal and very much popular method. Now talk about the transcervical techniques. Here, first the animal is restrained in a bale or a race in standing position. Then vulva should be cleaned with a towel and Vaseline should be applied to vaginoscope. That this is applied for introduction. This vaginoscope inserted in dorsal direction to the animal and cervix located to the gentle manipulation. Then the tip of insemination seat is guided towards the entrance of the uterine orifice and introduced with gentle rotating movements. Once the semen is deposited, it is advised to keep the female animal in the same position for 2 to 3 minutes, remain at rest. In young females, placing vaginal speculum or vaginoscopic is very painful. So in this case, only insemination pipette is placed in vulva and semen is discharged the bottom of vagina. So this is about the transcervical. Now discuss about the intrauterine laparoscopic techniques. These are the laparoscopic techniques in which the surgical incision is done and directly in the uterus the semen is deposited. These are the high-end equipments, and this is the laparoscopic tray in which animal is kept. Here we can see how the animal is inverted position and injections is giving through uterus region. You have to very skillful and identify the proper place. The animal should be restrained in 45 degree. For the laparoscopic AI purpose, endoscopic is performed by introducing an optical system in animal body by abdominal puncture. Here we can see then endoscopy is happening here. Then the AI is carried out by procedure known as laparoscopy. A small orifice is made with a trocar close to mammary gland and semen is inseminating through it. The insemination consists of injecting half the semen dose in each horn. Each horn should be half filled with semen straws. It is widely used in ovine species because there is low pregnancy rate obtained by the cervical insemination, around 20 to 35% due to difficulty in transposing the ovine cervix with its peculiar anatomical structures. Here, when we see endoscopically, this is the correct location of horns. Here we can see these are the two different horns and the injections are injecting into the horns. These are the experts doing insemination. So this is all about what we have talked about the artificial insemination. Lastly, we can conclude with the advantages of AI that it facilitates in progeny testing. We have already discussed the elite genetic population we were going to achieve, and it eliminates the unnecessary bugs also. The kids are obtained of desired need by the help of AI. The good record keep maintenance also at the farm is suggested. It permits the breeding time of unequal size. Semen can be easily transported across the globe, and it is very much useful method what we are currently talking, the artificial insemination in small animals.